we will start the Symposium 1, Animal Venom Peptides and Their Use. These sections, we will have Dr. Gisele Picolo from Instituto Butantan with the talk Snake Venom-derived Peptides in the Pain Management. And I would like to introduce Dr. Picolo. She holds a master's degree in experimental and comparative pathology and a PhD in pharmacology from the University of São Paulo, and an MBA in health innovation management. She is currently a level 4 scientific research at the Butantan Institute and director of the Laboratory of Pain and Signaling. She acts as principal investigator at the Center for Toxins, Immune Response and Cell Signaling, and at Technological Innovation Platform for Health Emergencies. She was awarded the IASP Developing Countries Collaborative Research Grant offered by the International Association for the Study of Pain in 2016 and was ranked among the top 5 best works on chronic pain at the 6th International Congress of Neuropathic Pain, Sweden, 2017. She was member of the National Organized Committee for the Brazilian Biology Olympiads from 2018 to 2024, and member of the Director Board of the Brazilian Society on Toxinology from 2015 to 2019, where she is currently member of the Council. Dr. Picolo will coordinate this section. Please, Gisele, be welcome.
Good afternoon, everyone. I would like first to say thank for Mariana Castro for this invitation. I'm from Butantan Institute, and I will show you this afternoon our main results about snake venom-derived peptides in the pain management. The therapeutic properties of natural products have been described for centuries, as well as the potential of several venoms as a rich source of molecules used for the screening of new compounds with potential therapeutic actions. And today, I will talk specifically about, results of the rattlesnake venom. Besides its toxic effects of this venom, which includes nephrotoxicity, neurotoxicity, myotoxicity, and others, several potential therapeutic effects have been described for the venom of the rattlesnake. The first reports about the analgesic effect of this venom date from 1934 and were described by Dr. Vital Brazil, which used this venom in patients with tumor process, and he observed that the administration of the venom induced both analgesia and tumor regression. These administrations was always done by parenteral route. Some years later, this effect was attributed to crotoxin. Crotoxin is the main toxin from the crotallid venom. It represents around 60% of this venom. And although its neurotoxic effect, it induces immunomodulatory, anti-inflammatory, anti-tumor, and analgesic effects in non-lethal and also sometimes in non-toxic doses. Para onde eu miro? Okay. Importantly, the administration of this toxin is always done by parenteral route, since cretoxin is not effective when administered by oral route. However, the crude venom induces analgesia when administered by oral route, which made us suppose that some other peptide could be also inducing analgesia in this venom. This effect of the crude venom was widely explored by our group. We published some articles about this analgesic effect, and in 2008, we described a peptide that was named cretalfin. Cretalfin is a 14 amino acid peptide with a disulfide bridge linking the cysteines in position 7 and 14, which induces analgesia, potent and long-lasting analgesia when administered by oral route. Interesting, at that time, it was a challenge to publish these results because the main question of the reviewers were why we were showing a peptide with analgesic effect by oral route. And it's today widely described. We have some other peptides that has effect when administered by these routes. Well, cretoxin is a heterodimeric protein. It's formed by a complex formed by two subunits. One subunit that is the phospholipase, named CB, and other subunit named cretaputin, or the subunit CA. And these two subunits are present in the individual snake venoms as a mixture of isoforms. Then we have already described 4 isoforms for CB and 4 isoforms for CA subunits that can form up to 16 natural crotoxins complex with relative differences in biochemical and pharmacological properties. And the cretalfin, the peptide that we described in 2008, it has the sequence exactly the same of the gamma chain of one of the isoforms of cretaputin. We explored this analgesic effect. We have many years studying these peptides. All of these papers are related to the analgesic effect of cretalfin, and we could observe that this peptide induces analgesia, a potent and long-lasting analgesia. The first graphic here, we can see analgesic effect of cretalfin under sensitization induced by prostaglandin A2. And we had the same results when we used carrageenan as an inflammatory agent. Cretalfin also induces analgesia in chronic pain models. Here we have the neuropathic pain induced by chronic constriction of sciatic nerve, and here a cancer pain model. And we can see that in all of these tests, we have a long-lasting analgesia, since this effect lasts from 2 until 5 days, depending on the model, with just one single administration. Besides the oral route, we can also observe an analgesic effect induced by this peptide when administered by intravenous or subcutaneous routes. And also, we couldn't observe the development of tolerance to this analgesia, which is an important side effect observed by some other classes of drugs, such as opioids. Also, cretalfin does not interfere with general spontaneous activity or locomotion of the rodents observed by open fields. The mechanism of this peptide involves the peripheral activation of CB2 cannabinoid receptors and also kappa opioid receptors, and also the release of dynorphin A, which is the endogenous opioids, the endogenous peptide which targets to kappa opioid receptors. Here we can see the release of dynorphin A in naive conditions, and here under inflammatory conditions. And in all of these graphics, we can see that the release of dynorphin A is completely blocked by the antagonist of CB2 receptors, showing that the release of endogenous opioids is a consequence of the first activation of CB2 receptors. Then we have that cretalphin first induce the activation of CB2 receptor that has a great cross-talking with opioid receptors, inducing the release of dynorphin A, which is the ligand of kappa opioid receptors that, once activated, stop with the conduction of nociceptive stimuli. We also observed that cretalphin is able to induce the activation of TRPA1 channels expressed in HEX cells. TRPA1 channels are channels involved in the pain conduction. And we observed that after a first stimulation of this channel, a second exposition of the component leads to a desensitization of this channel. I mean, the second answer is much lower, almost completely blocked in the second administration, showing a desensitization of these channels. We also evaluated the analgesic effect of this peptide in a model of autoimmune disease. Here, we used the experimental autoimmune encephalomyelitis model, which is a described model for multiple sclerosis, but in animals. In this model, the mice are immunized with MOG, which is a protein that is similar to a part of the myelin content, which is the protein that involves neurons. And then mice will have an inflammatory response both at peripheral and central sites, together with the production of antibodies against myelin. Then we have an attack in the neuron sheet of myelin and leading to a demyelinization. Then this is a demyelination model where we can analyze the clinical signs of animals through a table scores from 0 to 5, according to the level of paralysis of these animals. Here we can see that 4 days after the immunization, the mice showed a decrease in the pain threshold. And they show the sensation of pain. We injected cretalphin 1 day after the onset of pain. I mean, 1 day after the onset of the first sign of the disease, we injected cretalphin, and we can analgesic effect for the whole period of observation. Injecting cretalphin 1 day after the onset of pain, or even in 5 consecutive doses, I mean 1 dose by day, for 5 consecutive days after the onset of clinical signs, in both situations, we can see that cretalphin decreased the intensity of the disease. Then we removed the spinal cord of the animals at the peak of the disease and a period later on. We analyzed these spinal cords, labeling markers of microglia and astrocytes, and we can see that in both periods, I mean the peak of the disease and the later period, for both markers, we can see that the disease increase the activation of microglia and astrocytes, which is blocked by cretalfin in both periods. Also, cretalfin was able to decrease the release of interleukin A, interleukin 1, interleukin 17, and TNF-alpha, which are important pro-inflammatory cytokines at the central nervous system, which markedly contribute to the development of this disease. We also evaluated cretalfin in one model of neuropathic pain. We used a model named the partial constriction of the sciatic nerve. And in this model, we have 3 days after surgery, the decrease of pain threshold. That means the hypernociception of the animals. And in this model, we have a pain divided in 2 phases. The first phase, which is an acute phase that lasts until 7 days after the surgery, where this pain is easily blocked by non-steroidal anti-inflammatories. But 14 days after the surgery, this pain is not blocked by anti-inflammatories anymore. Then we have the pain, the chronic pain. And we can see that cretalfin may induce analgesia in both periods, acute and chronic phases of the pain that lasts for more than 24 hours. Here, instead of to label markers of glial cells, we treated the animals by intrathecal route with minocycline, that is an inhibitor of glial cells. And we saw a reversion of the analgesic effect induced by cretalfin. At this point, we have to think about it, because if I previously showed that cretalfin was inhibiting glial cells, and here I used an inhibitor of glial cells, I should have seen a potentiation of the analgesia. But I saw the contrast. I saw the reversion of the analgesia. How to explain that? Well, we know that microglia, as peripheral macrophage, are cells which can polarize to different states, M1 and M2 state. The M1 state of microglia is a pro-inflammatory state where it releases pro-inflammatory cytokines, and it's involved in the neuroinflammation. While the M2 state of microglia is a state which controls the neuroinflammation and is acting on the tissue repair. And in this stage of the microglia, we can see the release of anti-inflammatory cytokines and also analgesic components. Then we thought that maybe cretalfin could be inducing the polarization of microglia to a M2 state. To test that, well, the first point should be pro-inflammatory cytokines are decreased. We saw in the previous slide that interleukin 1, 17, and TNF-alpha were blocked by cretalfin. Here we tested interleukin 6, and again it was inhibited by cretalfin. The second point is, if microglia were polarizing to M2 state, it should be releasing analgesic components. To test that, we injected by intrathecal route anti-beta endorphin, anti-dynorphin, and anti-enkephalin, which were antibodies against the endogenous opioids, which bind to Mi, Kappa, and Delta receptors. And we could see that in all the situations, the antibodies blocked the analgesic effect, suggesting that these endogenous peptides were released in this model. Finally, to prove that cretalfin was acting polarizing cells, we used BV2 culture cells, which are a cell line of microglia. We incubated these cells with cretalfin for 24 hours, and we tested markers of M1 and M2 states of microglia by flow cytometry. And we observed here that cretalfin was able to decrease the polarization of microglia to M1 state stimulated by LPS, while it increased the polarization of cells to M2. As far as we know, this is the first animal toxins that make this shiftness of the state of the cell at the central nervous system. Well, using analyze tools to predict the ability of cretalfin to penetrate the blood-brain barrier and reach the central nervous system, we saw that cretalfin is not predicted to easily penetrate this barrier. Although we know that in chronic conditions, especially in inflammatory conditions, the permeation of this barrier is altered and some components may cross in these chronic conditions, we couldn't find cretalfin in the cerebrospinal fluid after route administration analyzed by mass spectrometry. Then these results suggest that cretalfin does not cross this barrier reaching the central nervous system, but this central effect may be due to the release of other components at periphery that may be opioids endogenous and other substances, and these substances are able to control the whole inflammation in these animals. Well, we got some really good results with this component, but unfortunately it was not developed as a drug to reach the market, to be clinically available. But considering the effects that we got, we used cretalfin, this peptide, as a leader molecule for the searching of lower sequences and analogs. Then, to identify the shortness sequence responsible for the analgesic effect, we did some analogs designed based on the first molecule, searching for essential amino acids. We removed the bridge, and we got finally a 5 amino acid peptide with a pyroglutamate at N terminus and amidation at the C terminus that we named mini-CRO. And this patent was applied this year in August. This peptide was designed in a collaboration with Denis Servan, a researcher from France. Comparing mini-CRO and cretalfin, we could see that even in lower concentrations, mini-CRO is able to induce analgesia when compared to cretalfin. Using the same concentration in the next assays, we could see that while cretalfin induced in this model analgesia for 72 hours or 3 days, with mini-CRO we can see effect for 6 days after one single administration. Both of them were able to control the neuropathic pain, and here again, as the cretalfin, mini-CRO is also able to control chronic pain with the lasting of 48 hours, 2 days. It does not induce alteration in general activity and locomotion of the rodents observed by open field, and again, it was not observed the development of tolerance to its analgesic effect. Then we started some assays of stability, trying to evaluate the degradation of this peptide in different conditions, and we tested different conditions of temperature and pH. And we see here incubating both mini-CRO and cretalfin for 24 hours in different temperatures. We can see that both of them are stable under different conditions. However, when we incubated these peptides in different pHs, we can see that while cretalfin is stable only in pHs of 7.4 and 8, cretalfin is stable from 1 to 9, showing that its stability under different pHs is greater than cretalfin. Well, as a conclusion, our data show the potential of an animal-derived peptide in the pain control. We could see a long-lasting analgesic effect greater than the most analgesics in the market. Comparing the concentration of the peptide with morphine, we could see that it's more potent than morphine, and no adverse effect is reported so far. In addition, its effect is preferentially peripheral, which reduces the chance of adverse side effects. Additionally, it presents a new mechanism of action when we compare to some other drugs, because we could see a polarization of microglia to AM2 state, and it can be used as a leader molecule for design of new peptides. Using this compound, we also could highlight microglia as a key player in the control of chronic pain behavior. And finally, considering the richness of our biodiversity, this data highlight animal venoms and toxins as a great source of new bioactive molecules with therapeutic potential. Here I would like to acknowledge my collaborators from Butantan Institute, from University of São Paulo, Hospital Sírio-Libanês, the Commissaria of Atomic and Alternative Energy from France, our financial agencies for the support, to thank to my whole group, the people from my lab, and you all, thank you for your attention.
Thank you, Gisele, for a very interesting talk. And now we are open for questions. Maria Helena.
Very nice, Gisele. Congratulations. I would like to know if you test the stability of your peptide with the plasma or serum, because you said that you did detect it in the brain, but perhaps it could be destroyed by the enzymes in the plasma.
Not yet. I have the samples collected, urine, blood plasma, and liquor again, but I didn't test it yet.
Yes, it's very interesting to be stable by oral via,
yeah.
But perhaps you don't find it in brain because it can be destroyed in circulation.
Maybe.
Because you test a peptide, antimicrobial peptide, that it's not effective, for example, to arrive to the lung, but when we modify this peptide, it's arrived and the bacteria was died. But when you test it in enzymes, find in the plasma, you note that the unpegylated peptide is destructed very fast, and the pegylated you can stay stable for 24 hours.
I do not believe that it is destroyed, because when we injected by intravenous routes, it induced the same analgesic effect for the whole period, for 5 days.
Okay.
The same way. Considering the properties of the peptide, it's not predicted to cross the barrier, the blood-brain barrier. That's why, because I think that it does not reach the central nervous system. But you are completely sure. I have to test it. I have the samples collected, but I didn't try it yet. I don't have all the results. I got just this one regarding the temperature and stability, and I don't have the other results yet.
But the strong argument you have if it has the same effect when they inject.
Yeah, and the lasting is the same for 5 days.
Very nice. And do you intend to develop it in clinical test?
Yeah, I really want to do with this peptide what we couldn't do with the first one. The first one was discovered in 2008. We had no experience. We lost time, and we couldn't do it in appropriate time. With this one, I would like to got it. My first intention is to make a formulation. That's the first step, because to perform the preclinical assays, I have to do it in BPL conditions, GPL conditions, and with not only the peptide, with the formulation. Then this is my next step. Try to work in this from the preclinical assays.
And this peptide, the crotalpine, exists in the venom, or it's a hydrolyzed product from crotapotina A, because you said that the sequence...
it exists in the venom per se.
But it's not a product from hydrolyzed?
Maybe too, because it's the same sequence.
Okay. Thank you. Very nice.
Okay. Questions, please? Where? Come.
Congratulations, Gisele.
Thank you.
It's a very nice presentation, learning a lot in this meeting. Thank you. I was guessing, you said that the peptide could polarize microglia from M1 to M2, but actually it doesn't cross the barrier. How can you imagine it's acting in the peripheral nervous system?
I couldn't find the first target of this compound. We did a lot of assays. We have several years of evaluation. We know its effect on TRPA1 channels. We know that induces the release of endogenous opioids. I suppose that induces the release of endocannabinoids, but regarding endocannabinoids, we couldn't see if the effect is direct or by indirect by this release. I know that it doesn't bind directly to any of opioid receptors, sodium, potassium, calcium channels, orphanin receptors, any other analgesic.
CB2?
No.
Because crotalpine acts in CB2 also.
It acts in CB2, but not directly.
Not there.
Yeah. And then I don't know which is the first target. We have a lot of suppositions, but I don't know. What we think is that the chronification of pain is a consequence of a peripheral injury. Then all the restructuration of the tissue starts at periphery, and then it is passed by the conduction of the signal to the central nervous system. I think that the first control is at periphery, and we have some or many anti-inflammatory mediators released at periphery, and maybe this one can be crossing from periphery to central nervous system, but I don't have this answer.
Is it easy to test this kind of cytokines peripherally? I don't know. I have no idea.
Cytokines we tested in blood samples.
Not in blood samples, but like in situ, in some kinds of nervous.
No, we only did in tissue. Pole tissue, where we have the nerve ends of the neurons, because the main source of endogenous opioids is keratinocytes that are distributed in the skin, and then we removed the skin, but together with the terminal of the nerve, and we evaluated the release both at blood samples and at tissue cells. We could see the release in the skin.
Great.
It's just one step. It's
yeah,
not the
very nice,
answer.
Exciting. Thank you.
Thank you.
Gisele, that's a great presentation. Congratulations again. Trying to bring these venoms into a more significant stage of development has been challenging, but it's great to see that great advancements have been done. I'm interested on on your focus on the penetration of the BBB. I know crotaalfin does not penetrate, but your mini peptide does, right? You don't know yet.
I don't know yet.
But when you create that mini peptide, was with aim to create an oral deliverable, right?
What? Say again.
When you make this smaller version of crotaalfin, you are hoping to have a oral available version of crotaalfin. Is there any, have you considered using the approach used by the pre-out? Because pre-out is administered intrathecally with an osmotic pump, and it's FDA approved in US and Europe. So crotaalfin probably would fit well with that pipeline of delivery, and with the kind of delivery you would also avoid the digestion of the protease destruction of these peptides in the periphery. Have you considered that?
Yeah, I tried crotaalfin by intrathecal route. It does not induce analgesia when we tested Intl flick tests and hot plate tests. But when we injected, when we tried some model of inflammation, we can see analgesia. But we cannot see in models that does not involve some kind of sensitization.
So crotaalfin, it does have analgesia only in inflammation-associated pain, but it's not neurogenic. It is only the inflammatory. Okay, well, that clarifies that. Thank you.
You're welcome.
Congratulations for the beautiful work. Did you test the mini-crow for the shift in microglia from M1 to M2?
Not yet.
And did you test either one of the two in other cells that also have similar patterns like macrophages or neutrophils?
No, no. We started the tests of using cells. My first point was to see if it was cytotoxic for neurons. And then I invested first in neurons to be sure that it's not cytotoxic for this type of cells. Then we tested in microglia. We had tested my crotaalfin in microglia. I started mini-cro in neurons. This one I know that's the same effect. It's not cytotoxic. But I didn't perform all the tests with this short peptide. Not yet. I didn't try it. Not yet.
Thank you. Because this is an amazing feature of the. We are all looking for something that can shift the cells.
That can control the inflammation with our endogenous machine, controlling the development of the chronic pain. If we can avoid the development of pain, we can, because chronic pain is so difficult to be managed. Then if we have some type of drug that can control this development using our own machine, releasing just our endogenous substance, would be great. Yeah, that's my intention. Now I would like to call our next speaker, Dr. Guilherme Brand. Guilherme holds a degree in biological science from the University of Brasilia and a PhD in animal biology with an exchange period at the University of Glasgow from 2006 to 2007. Worked as an analytical biochemistry at Sarah Network of Rehabilitation Hospital from